Hi, my name's Steve Bell, and I'm a professor of biology at MIT and an investigator of the Howard Hughes Medical Institute. And what I'd like to tell you about today is how we've used single molecule biochemical studies to understand a key event in the initiation of eukaryotic DNA replication. I first want to start, however, by reminding you that single molecule studies are not new to DNA replication. As early as the late 1960s, Huberman and Riggs used the incorporation of radioactive nucleotides to visualize individual replicating DNA molecules. And using this analysis, they were not only able to identify the fact that eukaryotic chromosomes have multiple origins of replication, but they also were able to demonstrate that those origins led to the initiation of bidirectional replication. And ever since this key seminal observation, it has been an important question in the field of DNA replication, how the bidirectional nature of DNA replication is established. And that'll be a theme of my presentation today. Now, as I discussed in the previous part of my talk on initiation of replication, the initiation of eukaryotic DNA replication is segregated into different parts of the cell cycle. The first event of helicase loading, which marks all potential origins of replication, occurs during the G1 phase of the cell cycle. The second event of helicase activation can only occur when cells enter into the S phase of the cell cycle. And this is triggered by the action of two kinases and leads to the formation of the activated helicase and eventually the replisome. And again, if you want more details about this, you should look at the previous presentation on the initiation of replication. For this presentation, we're just going to focus on this earliest event of helicase loading, which I'll remind you marks all potential origins of replication. So I want to give you a little bit more detail about this event. So this event requires opening of the heterohexameric ring-shaped MCM227 complex, placing it around the double-stranded DNA, and then closing it again such that it encircles that double-stranded DNA. And importantly, this occurs not once, but twice, such that you assemble an MCM227 double-hexamer with each of their N-terminal ends, which are at one end of the protein, interacting with one another, as is illustrated here. Now, these are arranged in such a way that the helicases, once activated, will leave the origin in opposite directions, and thus this is the first marker of the bidirectional nature of DNA replication. Now, the MCM proteins don't do this alone. They require the action of three additional proteins called helicase loading proteins, called ORC for origin recognition complex, CDC6, and Cdt1. Now, a variety of studies from a number of labs has led to an early understanding of this process, and that's outlined on the next slide. So we know that the first step in this process is the recognition of the origin DNA by the origin recognition complex, which binds a specific sequence found in the yeast DNA replication origins and binds to other attributes of origins that don't have specific sequences associated with them. When cells enter into the G1 phase of the cell cycle, ORC is joined by CDC6 to form a six-membered ring around the DNA, and then this can in turn recruit the MCM227 Cdt1 complex to form an initial OCCM complex with the DNA. Now, OCCM stands for ORC, CDC6, Cdt1, and the MCM227 complex, so all four proteins are present at this point. Now, at the time we started this studies, there were a lot of questions about what happens after the formation of the OCCM, and this was in part due to the type of assays that were used to monitor these events. They typically involved attaching magnetic beads to many copies of origin-containing DNA, and then to those origin-containing DNA attached beads, you could incubate the four purified proteins, ORC, CDC6, Cdt1, and MCMs, with the DNA, and then after various times of incubation, you could monitor the association of the proteins with the DNA by simple washing away of non-associated proteins. Now, this kind of approach worked very well to understand the first requirement for any of these factors. So, for example, you could leave ORC out of the reaction and none of the other proteins would associate, indicating that it acted at an initial step. In contrast, if you left Cdt1 out of the reaction, you get the ORC binding, you get ORC-CDC6 interaction, but you never got the OCCM. The problem with this approach is that it's very difficult to look at downstream events, and there's two reasons for this. One is if a protein's used twice, leaving it out only reveals the first step. And the second is if you're looking, for example, at the release of a protein, unless that happens very synchronously in the reaction, it's very difficult to monitor that in a sort of a bead-based assay. Indeed, these sorts of multi-protein assembly events typically do not occur in a highly synchronous nature in a so-called ensemble reaction, where you're adding many, many copies of the protein, somewhere around 10 to the 12th to 10 to the 15th copies of the protein, and a similar number of DNA molecules. The odds that any event is going to happen within a narrow window of time is essentially zero. So we were interested in understanding the events that occurred after the OCCM, and in order to do this, we've turned to single molecule studies. And the key questions we were interested in answering were, for example, what are the fate of the proteins in the OCCM complex? They're not part of the final double-hexamer that's formed on the DNA that requires ATP hydrolysis to occur, and it involves multiple steps. But we don't know when they leave, and is there any, for example, order to their departure. Secondly, we wondered how many copies of these proteins were required to load 2 copies of the helicase. Was it 2 copies of all the proteins, or 2 of some, 1 of others, or even possibly 1 of each of the proteins? And finally, perhaps the most important question was, how do you assemble a double-hexamer that has 2 copies of the helicase in opposite orientations? So what's the mechanism of loading it once in one orientation and then the second time in the opposite orientation? So as I said, we turned to single molecule studies to address this. And I'd like to take just a little period now to explain the advantages of single molecule biochemistry and why we thought this was a good use of our time to pursue. So first, before I do that, I should acknowledge who the we is. So all the studies I'm going to tell you about were done by a very talented former graduate student, Samina Takao, and they were all done in close collaboration with Jeff Gellis's lab, and particularly with Larry Friedman. And not only did they build the microscope that these experiments were done on, but they were integral to all the analysis of the data that Samina generated. Okay, so why single molecule studies? First, because you're constantly monitoring the protein associations with individual DNA molecules, you can see dynamic or unstable interactions that are very difficult to see using the bead-based assays. Because the bead assays take 2 to 3 minutes to wash away the unbound proteins, any association that lasts less time than that is essentially as if it never interacted at all. Secondly, you can determine the precise number of proteins that are acting on an individual DNA molecule. Now, while in a bulk assay you can get an average number, so for example, you could determine if you have a picomole of DNA and you have a picomole of protein that's bound to that DNA, that on average there's 1 protein per DNA molecule. But I'll point out, if you have a less than efficient reaction, you could have half the molecules having 2 molecules bound and the other half having none, and you'd see the same average. So you can't determine a precise stoichiometry. And finally, and perhaps most importantly, the ability to synchronize the reactions artificially is a huge advantage of single molecule studies. As I mentioned earlier, the bulk assays are intrinsically asynchronous. But in a way that I'm going to describe next, you can use artificial synchronization to create a synchronized set of molecules and get very detailed kinetic understanding of the reaction. So let me just illustrate that for you. So in the context of a bulk assay, you would have many DNA molecules, and you would add various proteins here, a blue circle and a red triangle, and you'd allow them to interact for some period of time. And then at the end of that time, you would wash away the unbound proteins, and you would be left with essentially what's illustrated here. Now, there's 2 things you wouldn't know. First of all, you wouldn't know the history of any of these molecules. In other words, did this blue molecule here, for example, bind and then come back on again, bind, release, and come back on? Did it bind once? Did this red triangle ever bind to that blue circle? You won't know those things. In addition, short of just leaving out a protein, you don't even know what the requirements for 2 different proteins are in this process. Okay? But most importantly, you have no kinetic understanding of how this happened. You just know the endpoint. Now, if you do the same experiment and monitor the events on a single molecule level, they'll still be asynchronous, as was illustrated before. So this is time on the x-axis, and the proteins bound more or less on the y-axis. And you can see now, we see the history of these proteins. We can see at time 0, neither DNA is bound, right? And then later on, we see a blue circle bind, and then a blue circle and the red triangle, and then we see that the red triangle comes back off again. Now, if we use these time frames that I've illustrated here, these events would be occurring at very different times. But we can take and artificially or computationally synchronize these events. For example, we could say we're going to start timing when the first blue circle gets on the DNA. And that's going to change everything, because now we can look at the time from the blue circle arriving to a red triangle binding. And this is likely to be much more constrained than the bulk reaction, where we're going to have many of these events starting at different times and therefore the second event occurring at different times. And we could also, for example, ask how long does it take for the red triangle to fall off? So we start with the blue triangle, the blue circle and the red triangle, and then we measure the time until we just have the blue circle. So this gives us an ability to get kinetic understanding of complex biochemical reactions that's essentially inaccessible by conventional bulk assays. So how do we convert a helicase loading assay into a single molecule format? So what we've done is to simply exchange those magnetic beads for a microscope slide. And when we do this, we simply tether origin-containing DNA to the surface of the slide, and then at the other end of the DNA, we place a blue fluorophore that we can monitor by microscopy. Then we perfuse into that chamber, that microscope chamber, all the proteins required for helicase loading, at least one of which, is labeled with a different colored fluorophore. Although I illustrate here that with two different colors, so a red and a green fluorophore, green on CDC6, red on the MCM proteins. So at this point, we use something called TIRF microscopy, or total internal reflection microscopy, in order to create what's called an evanescent field of illuminated molecules. And this evanescent field, importantly, only stretches about 100 to 200 nanometers above the surface of the slide. So only molecules that are in the region that the DNA occupies can be monitored. Any molecule above the evanescent field will not be fluorescent, because the illumination will not reach that point. And this is very important because it allows the use of much higher concentrations of protein, because most of the proteins are not illuminated, and that gets rid of lots of background. Now, how do we monitor DNA binding? We use a simple proxy for DNA binding, which is the co-localization of the protein fluorophore and the DNA fluorophore. And this, in this particular assay, it indicates DNA binding. And indeed, this process, which was originally developed between the Gellis lab and Melissa Moore's lab, is called co-localization single molecule spectroscopy, or COSMOS. So how do we analyze this data? So this is an image of DNA molecules that are attached to the surface of the slide, or a field of view of those DNA molecules. And what we do is we identify individual DNA molecules, and then we monitor not the DNA fluorescence, but the fluorescence of one of the proteins. So we can ask over time, how does the fluorescence co-localize with each individual DNA molecule? So there's about 400 DNA spots here, and you do this 400 times. You can then create traces of the data in which you can see that, for example, in this trace, the MCM proteins are first not associated. This is the baseline here. And then you see two short associations. And finally, over at the edge here, a long association of MCM proteins. Okay? And so we can do this for each of the DNA molecules and determine a time of association for each one and a length of association. Okay, so that's how we analyze it. How do we know it's actually reflecting helicase loading? So I'm going to show you one control. These are two movies watching the association of the MCMs with DNA molecules. You're not going to see the DNA molecules, but take my word for it. They are associating with the DNA. And I've done one movie in the absence of CDC6 and the second movie on the left in the presence of CDC6. And what I think you can clearly see is when CDC6 is present, you see an accumulation of MCM molecules throughout the slide, whereas in its absence, you basically see a few smudges of probably denatured protein that are sort of hanging out on the surface of the slide. Now, I've shown you this control. I'll tell you that not only does this control work, but if we leave out ORC or origin DNA, or we prevent ATP hydrolysis, all of them inhibit this, and these are all hallmarks or characteristics of helicase loading that's been determined in other types of biochemical assays as well as in vivo. So, we have an assay. How do we use it? So the first thing I want to address is the stoichiometry between the origin DNA, the helicase, and helicase loading proteins. So to do this, we took advantage of the ability to label two different proteins and monitor the DNA at the same time. And we focused initially on CDC6 and Cdt1. And the reason for that is that we knew CDC6 and Cdt1 were essential for MCM loading. Okay? However, in bead-based assays, the association of these proteins was essentially not detected unless we inhibited ATP hydrolysis and stopped the reaction at this OCCM step of the reaction. Otherwise, it was as if they were never there. And we knew they had to be there because they were essential for the reaction. So we thought the single molecule setting would allow us to monitor these events and get an idea, for example, of how many CDC6 molecules does it take to load 2 MCM hexamers. So we did this again by labeling either Cdt1 and MCMs or CDC6 and MCMs with different colored fluorophores, so we can monitor both molecules at the same time. I'll first show you the example with Cdt1 and MCMs labeled, and then I'll show you an example with CDC6. So I've shown in this left-hand panel, you can see traces of Cdt1 association with DNA in green and MCM association with DNA in red. And we've shifted the baseline of the MCM trace up so that you can see both traces simultaneously in the plot. The first thing I hope you notice is that the MCMs associate with the DNA in a one-at-a-time fashion. We see an increase of fluorescence here and then a second increase where the second arrow illustrates. Now this could be because we have 2 MCMs loading in the first step and another 2 in the second step. However, we can eliminate this model and demonstrate that it's actually 1 MCM and then a second MCM using a process called photobleaching, which stochastically inactivates fluorophores. So we can take, at the end of the reaction, we can turn up the lasers so that we see photobleaching, and this, because it's a stochastic process, if there's 2 fluorophores, the fluorescence will decrease in stepwise fashion in 2 steps. If there are 4 fluorophores, we'd see 4 steps. And Samina's done many of these experiments, and we always see a 2-step loss of fluorescence, indicating that this is a first and a second MCM protein. The other thing I think you'll clearly see is that the time of Cdt1 association is identical to the time of the MCM association. However, Cdt1 comes off rapidly, leaving behind the MCM molecule. So you see a Cdt1 associated with the first loading event and then a Cdt1 associated with the second loading event. But both of those are much more short-lived than the corresponding MCM association. You can see that this is indeed occurring simultaneously in these 1-second frame shots of either the MCM or Cdt1 fluorescence, and you can see both of them occur within a 1-second window of each other getting on the DNA. And we know this is actually true at higher time resolutions as well. So that's Cdt1. Clearly we need 2 molecules. This doesn't turn out to be surprising because we know Cdt1 and MCM form a complex. What about CDC6? Well, we see a very similar pattern. You can see, again, we have a first and a second MCM binding to the DNA, and each one is associated with a CDC6 molecule getting on at the same time, but coming off rapidly thereafter. The only difference, and this is consistent with our previous biochemical studies, is that CDC6 always gets on the DNA before the MCMs get on the DNA, usually by at least 10 to 15 seconds. Okay? So if I summarize this, what I would tell you is that this shows that you need that distinct Cdt1 and CDC6 molecules are associated with loading the first and the second MCM complex. Now, one thing we wondered about is whether the release of these molecules might be ordered. if it was ordered, it might suggest that this is connected to the process of helicase loading, because that would suggest that there's some need to keep one CDT1, for example, after CDC6 or vice versa. And you can see already that the synchrony of these reactions is, in this case, not bad, but not great, especially if you look at the second loading. So we use this post-hoc synchronization in order to synchronize the events according to a defined event. In this case, we chose the arrival of MCM on the DNA. So whether we're measuring CDC6 release or the CDT1 release, we measure the time with a starting point of t equals 0, so to speak, of when the MCM that they're associated with bound to the DNA. And then we asked how long until the CDT1 releases versus the CDC6 releases. And we plotted this in the form of a so-called survival curve. And this just shows what percentage of molecules are still associated with the DNA at various time points after MCM binding. And I think you can clearly see that CDC6 on average releases well before CDT1. Now this is done with two different experiments in comparing the results, but I'll tell you that when we label CDC6 and CDT1 in the same reaction and look at their release from individual DNA molecules, we see exactly the same result. CDC6 releases before CDT1. So this suggests that those release events are actually coupled to some important element of the reaction. So we also wondered, is this the same for the first and the second MCM molecule? So we did the same measurement, and we segregated the first MCM loading event and its associated release of CDC6 and CDT1 and the second one. And the clear outcome of this is that whether you're looking at CDC6 in blue on the left or CDT1 in red on the right, the first molecules are always released approximately in half the time that the second molecules are. And this gave us the first hint that maybe the loading of the first MCM doesn't work in exactly the same way as the second MCM. Okay? So let me summarize what I've told you so far. So we know that you start with this OCCM complex, and we end with the double-hexamer, but now we know a lot of events that occur in between. We know that we have an ordered release of CDC6 and then CDT1 from the DNA, and then we have an ordered assembly of a second CDC6, a second MCM 2 through 7 CDT1 complex, followed by another round of CDC6 and then CDT1 release, albeit a little slower. Now, you'll note at this point that while we've gained a lot of understanding of CDC6 and CDT1, there are two questions we haven't addressed. One of them is the key question of how you make a head-to-head double-hexamer. How do you load one MCM in one orientation, the second in an opposite orientation? And the second, which turns out to be related, is how many ORC molecules are required for this process. Now let me explain why they're related. So there are two basic models to how you can load two MCMs in opposite orientations. In the top model, would suggest that you do the same reaction twice with two different ORC molecules that are bound in opposite orientations to one another. In the first reaction, you use the first ORC molecule to bind, as I described before, to the C-terminal region of the MCM complexes, and then that's loaded, and then at some point you have a second ORC molecule bound, and it performs the same reaction just in the opposite orientation. These two MCM complexes then come together and form the double-hexamer. A different model would suggest that there's only a single ORC molecule on the DNA that does the first round as we've described before, but that the second round is mediated not by interacting with ORC, but by interacting with the first MCM protein using this N-terminal interaction that's part of making the final double-hexamer. And I should point out that the double-hexamer is very tightly held together. So what I think is clear is that one key difference between these two models is the number of ORC proteins that would be present on the DNA during the loading of a double-hexamer. And so Samina did the same types of experiments I've described for CDC6 and CDT1, but using a ORC molecule and MCM labeled instead of either CDC6 and MCM or CDT1 and MCM. And this just shows two sample traces for those experiments. Again, you can see the first and the second MCM being loaded, but this time we see a single ORC molecule, a single increase in the ORC-associated fluorescence that lasts throughout both the first and the second loading events. And I can tell you, but won't show you, that photobleaching experiments, again, demonstrate that this is one ORC molecule, not two ORC molecules binding the DNA simultaneously. What I think you'll also notice is that while ORC stays on the DNA after the first loading event, it's rapidly released after the second loading event. And this suggests that the ORC release might be connected to completion of a double-hexamer assembly. And we wondered whether this might be temporally connected to some of the release events that we looked at earlier. So we again determined the time of ORC release relative to the arrival of the second MCM, okay, because after the first MCM it stays on for a very long time. And what I think you can see is that it's clearly distinct from the blue line, which is the CDC6 release events, but it sits pretty close to on top of the CDT1 release events. And in fact, we can statistically show that these are not significantly different events, suggesting that the release of ORC is coupled to the release of CDT1 from the DNA. So this suggests that the one ORC model is correct. And in order to address that more, Samina turned to something called fluorescence resonance energy transfer. And so what she did was to label one population of ORC with a green or donor one population of MCM proteins with a green or donor fluorophore, and a second population with a red or acceptor fluorophore. And it turns out you can use these two as a molecular ruler, and when the double hexamer comes together, you will see a different level of what's called fret than you will when they're apart. So let me illustrate that here. So when they're apart, when you illuminate the green fluorophore, it will generate an emission that has the potential to activate the red fluorophore. However, if they're far apart, most of that emission will not hit the red fluorophore. In contrast, you will see what's called fret when they're within approximately 10 nanometers of one another, and then when the green fluorophore emits, it will actually excite the red fluorophore. And so even though you're exciting only the green fluorophore, you'll still see red emissions. And in fact, the telltale sign of this is the green emissions go down while the red emissions go up when you're in a fret state. Now importantly, when Samina set this up, she picked sites that we knew were close together in the double hexamer to modify, and she could demonstrate whenever she saw fret, it had the hallmarks of a loaded double hexamer. So the real question we wanted to address is, when does this happen? If the model that we proposed is correct, this should be a very early event in forming the double hexamer. We don't know that this particular region will be the first place that comes together, but it should come together relatively quickly. And so we measured the time of fret formation relative to these other release events after the second MCM. Recall, we can't form fret until we have 2 MCMs on the DNA, so we can't do any measurements after the first one, but it also wouldn't be relevant. Okay? You can see the ORC and CDT1 coming off later than fret formation, and I should point out that the fret formation, we have an inverted axis. So as it goes down, more and more molecules have fret. Okay? So you can see in the green line on the far left, that's the earliest event we can see. In some cases, it's instantaneous upon the initial association. In other cases, it takes more than a few seconds to occur, but it's always before CDC6, and it's always before CDT1 release, consistent with the model with this being a very early event in the recruitment of the second MCM molecule. Okay, so let me summarize what I've told you so far. Our original understanding was this initial association event where we had ORC, then CDC6, then MCM CDT1, and we formed the OCCM, and then there was a lot of question marks, and we only knew there was ATP hydrolysis. Now we have a much better understanding. We know the first event after the OCCM is this ordered release of CDC6 and CDT1, and I'll tell you but didn't show you that we know the ring is in fact closed at that point. Then we have a second CDC6, a second MCM CDT1 binding, and this leads to the formation of the first double hexamer interactions, and we believe that's mediated by the N-terminal interactions between MCMs, and that's in turn followed by the release of CDC6, and we believe the simultaneous release of CDT1 and ORC from the DNA. And there's several properties of this model that we like in terms of driving the formation of a double hexamer of DNA, a double hexamer of MCMs on the DNA. So one is that because ORC stays bound after the first MCM is loaded and then is only released after the second MCM is loaded, you will drive the reaction towards double hexamer formation rather than having the possibility of having single hexamer interactions. And secondly, I hope it's obvious by now, by using the N-terminal regions of the two MCMs to recruit the second molecule, you will ensure that you always form the head-to-head double hexamer at the origin. Clearly, there's much more that can be done using single molecule studies to understand the events of replication initiation. For example, we can start incorporating mutant proteins into this process to understand when key events such as ATP hydrolysis or key interactions occur and how they influence this process, especially kinetically. In addition, we're very excited to use FRET to monitor additional protein-protein interactions. I showed you using it to monitor the formation of the double hexamer, but you could also use it, for example, to monitor the opening and closing of the MCM227 ring during the loading process, which would be one of the most key events in understanding loading. Finally, much of the replication process has been reconstituted with purified proteins, and so extending this same approach to look at the helicase activation and replisome assembly process is a key goal for my laboratory. So I'd like to end by acknowledging our collaborators. As I said at the start, all of this work was done in close collaboration with Jeff Gellis's lab, as well as Larry Friedman, and they, as I said, are integral to the analysis of all the data, as well as building the microscopes that allow this kind of research to be done. I'd also like to thank the Levis lab at the Genelia Research Campus, who developed a number of long-lasting fluorophores or photostable fluorophores that were essential for the success of this research. And finally, I want to thank the members of my lab. Most of the work was done by Samina. However, she was assisted by Nicola Iveka, who labeled many of the proteins, which can be one of the most challenging parts of these studies. And finally, I just want to thank the funding sources, the National Institutes of General Medical Sciences, as well as the Howard Hughes Medical Institute.